I have conducted 341 post-mortem examinations over the course of my career. I have cataloged the dead of 11 registered species. I have opened bodies that had been dead for an hour and bodies that had been dead for a standard week. I know the smell of autolysis across a wider range of taxa than anyone else currently employed by the Survey Division's Xenobiological Mortuary Service, and I do not say that with pride. I say it so you understand that when I tell you the human body alarmed me, I am not speaking from inexperience. The subject arrived on the third day of our transit back from the Lethus margin. It had been recovered from a surface site by the forward team, confirmed deceased at point of recovery, sealed in a standard stasis-grade preservation envelope, and transferred to my examination suite with a routine intake notation. Adult human, female, approximate age 40 standard years. Cause of death: blunt force trauma to the cranial vault. Stasis duration prior to examination: 11 hours. Estimated time of death prior to stasis induction: 2 to 3 hours. Total elapsed time from death to my table: 13 to 14 hours. By the numbers, I was expecting very little. For 11 of those 13 hours, the body had been held in a low-temperature preservation field that arrests most biological activity in registered species within the first 40 minutes. 2 to 3 hours of unassisted post-mortem time before the field engaged. That is a narrow window. For every species in my registry, the tissue I receive after that kind of stasis history is quiet. The internal chemistry has wound down. The cells have begun their orderly disintegration. There is nothing to measure that surprises you. I made the first incision at the 4th hour of my shift. The first thing I noticed was resistance. Not the resistance of rigor. I know rigor. Its timing curves, its regional variation by species and ambient temperature. This was different. The musculature of the upper abdominal wall yielded to the blade correctly, but the underlying fascia had a tensile quality I had not encountered before. It was not rigid. It was, I will use the word that I used in my notes at the time, before I knew enough to be embarrassed by it. It was alert. The tissue had a quality of structure that I associate with a body that is still doing something. I noted it and continued. The second thing I noticed was the temperature gradient. Our preservation field had held the subject at 4 degrees above Celsius 0, which is our standard archive temperature for soft tissue preservation. Every registered species equilibrates within 2 hours of stasis induction. The body cavity, when opened, should have been uniform, 4 degrees throughout, top to bottom, organ to skin surface. The human body was not uniform. The core temperature, at the liver bed, was 6 degrees above my instrument's baseline. The thoracic cavity read 5.8. The abdominal cavity read 5.4. There was a gradient. 13 hours after death, in a body held at 4 degrees for 11 of those hours, there was a measurable directional temperature gradient from core to periphery. I checked my instruments. I recalibrated. I checked again. The gradient held. I summoned my junior cataloger, F. Drask, and asked him to verify my readings independently. He did. He looked at me and said nothing, which is the correct response when the data is telling you something you do not have a framework for. I went back to the tissue. The autolytic enzyme profile was wrong. Autolysis, the self-digestion of cells after death, when the enzymes that the cell used for normal metabolism are released into surrounding tissue because the mechanisms that contained them have failed, follows a predictable curve in every registered species. The curve is steep at first and then levels as the substrate is consumed. At 13 hours post-mortem, with 11 hours of stasis interruption, I expected the autolytic curve to be nearly flat. The human body's autolytic enzymes were running at a rate I would associate with a body dead for 4 hours, unpreserved, at ambient temperature. The stasis field had slowed them, yes, but they had not stopped. They had resumed. The cellular machinery was still turning, still consuming, still processing, as though 13 hours were not enough time for the body to accept its own condition. I called threat analyst Sorette Davin at the end of my 2nd hour of examination. She arrived at the suite in 15 minutes. I showed her the temperature gradient first. She studied it for a long moment without speaking. Then I showed her the immune residue profile. This is the finding that I think about most, even now. In any species, the immune system's active agents, the proteins, the complement cascades, the residual cellular components of the inflammatory response, begin to degrade immediately after death. Without active circulation to carry them, without the metabolic energy that maintains their structure, they denature, they lose shape, they become inert. In a body 13 hours dead, I expect to find immune residue the way I expect to find a language, the shapes of words, but nothing speaking. The human immune residue was not denatured. The complement proteins in the soft tissue of the upper chest and the peritoneal cavity were structurally intact, not partially intact. Intact, in the sense that if I had introduced a pathogen sample to that tissue, the immune proteins would have responded to it. 13 hours after death, in a body that had spent 11 of those hours in a stasis field, the human immune system was still, in any meaningful functional sense, armed. Davin looked at the readout for a very long time. "What is the half-life on these proteins in a living subject?" she asked. I told her. Complement proteins in living humans have a half-life of approximately 18 to 24 hours, replacing themselves continuously through active synthesis. And without active synthesis, she said, without circulation, without metabolic support. I told her that the degradation kinetics in post-mortem tissue, absent stasis, should reduce complement protein activity to near zero within 6 to 8 hours of death for most species. She looked at the readout again, and the stasis field. I told her that the stasis field should have arrested the degradation process in its tracks, preserving whatever state the proteins were in at the time of induction, which, in a body dead for 2 to 3 hours, should have left them at perhaps 40 to 50% of living functionality, declining. She tapped the readout. "These are reading at 81%. I said yes. She said nothing for a moment, then, "Run the gas signature." I had not yet completed the post-mortem gas analysis, the measurement of the internal gas composition, produced by bacterial and cellular decomposition. I ran it while she watched. It took 9 minutes. The result came back with a species flag I had never seen triggered in a post-mortem context. The flag reads, "Profile does not match any catalog species in post-mortem state. It is a flag designed for first-contact living specimens whose baseline physiology is unknown." It had never, in 341 examinations, triggered on a dead subject. I looked at the flag. I looked at Davin. She was reading the gas breakdown on her own screen. "The bacterial load is wrong," she said. "Look at the composition." I looked. The decomposition bacteria present in the human gut and tissue, the organisms that, in any species, begin their work the moment the immune system ceases active suppression, were present, yes, but their population density was lower than it should have been at 13 hours. Significantly lower. The immune residue was still suppressing them. A dead body, immune proteins still active enough to retard its own decomposition. Davin set her screen down on the edge of my examination table. She looked at the subject for what I can only describe as a careful moment, the kind of careful that is not reverence exactly, but is adjacent to it. "How many living humans do we have in the registry?" she asked. I told her the number. "And how many of their biology teams flagged the immune complement retention figures in post-mortem tissue?" I told her I did not know. Post-mortem data is not a routine priority for first-contact xenobiological surveys. You catalog the living. The dead are assumed to be finished. Davin picked up her screen. "Amend your intake protocol," she said. "Any civilization handling a human body for the first time, as cargo, as recovered remains, as a specimen, needs to know what they are handling." She paused. "A body that has been dead for 13 hours and is still suppressing its own bacteria is not an inert object. It is not a safe object. Anything that interacts with that tissue expecting the biology to be done is interacting with a system that has not been informed of that expectation." She left. I stood over the subject for a long time after that. I finished my examination. I completed the catalog. I wrote my report with the care I give every report, and I submitted it through the standard channel with a Priority 1 flag, which is used for findings that require immediate policy review. In 31 years and 341 examinations, I had never used that flag before. I have thought about it many times since, about what it means to catalog a species whose dead are still, in any biologically meaningful sense, active, about what it tells you about the living, if the dead carry that much still forward into the silence. The living, I concluded, must carry something we do not have a category for at all.